Okay, all right, let's get started. Today, we are going to have Professor Balaram from Bangalore talking on GN Ramachandran and his impact on protein conformations. I know Balaram since 1978 when I joined IISc Bangalore. He was already in the biophysics department. He did his undergraduate education at Pune University, Ferguson College, and then he did his master's at IIT Kanpur, where his teachers were almost who in the Indian science scene. And in fact, those are the people who changed the science infrastructure in India. So like Professor C.N.R. Rao, P.T. Narasimhan, George Ranganathan, and D. Balasubramaniam, and many others. So they are all his teachers for MSc. And then he did his PhD at Carnegie Mellon University with Bhatner B, an NMR expert. And I was just looking at his publication in JAX for his PhD. He did the first work on negative nuclear overhauser effect, and also the first paper on intermolecular nuclear overhauser effect. And those are the techniques which has become very popular for mapping out host-guest structures. And then he did a postdoc. Okay, I'm sorry, I'm going to take a little bit time to tell about Balaram because I don't get the opportunity many times. So I will just use it. So he did his postdoc with Woodward at Harvard for a year, and then he returned to India, IISc Bangalore. So where he joined the same department where G. Ramachandran was setting up the department. So he joined as a lecturer in 1973. So since then he has been assistant professor, associate professor, professor, dean, and then dean of divisional dean, and eventually he became the director of the institute, the highest job in the institute. And the institute is probably the very best in India. So it is not that easy to get to the director. I just want to tell the students. So, and then in 2014, he retired from the directorship, and then he became a professor at NCBS, National Center for Biological Sciences. So with respect to his awards, he has got numerous awards. I will tell you only two. The first one is Bhatnagar Prize, which he got in 1986. And the latest one, that is this year, he got the American Peptide Association Award, Bruce Merrifield Award. So he just got it maybe a few months ago. He has got over 400 publications, and then probably he has trained more than 100 students and postdocs. I don't have his CV, so I'm just looking from things which I collected and also from what I know of him. So during his time at the Institute, he has done work on the peptide conformations, which I saw when his lab used to be across my lab. So I knew pretty well his lab, and I used to use his fluorimeter and absorption spectrometers. All these things I used to use in his lab. And then with respect to peptides, he worked with the NMRs and IRs, CD, mass spec, and X-ray crystallography. In the X-ray crystallography, he collaborated with Isabella Carl, who is extremely well-known crystallographer. Her husband is the one who got the Nobel Prize, although there are lots of people who say she should have got the Nobel Prize along with him. And he has been on the fellowship of various academies in India, and then also Third World Academy of Sciences. And one of his crown achievement is being the editor of Current Science. Current Science in my days was an almost unknown journal, or it is. But then he and Professor Ramachandran, who was the director of the IISc at that time, they resurrected the journal. And then Balaram's editorial is very popular. People read the journal for the editorial more than for the articles inside. So the people were always waiting for it is like waiting for your magazines to Time Magazine or New Yorker or something to come. So people wait for this week to look for this editorships. He has got more than 300 editorials over a period of 10, 15 years. And he's on the editorial boards of lots of journals. And I'm already going over my time limit. So I will tell you a couple of small things. Well, it is more of a administrative jobs. He's on the Science Advisory Council to the Prime Minister of India. He is on the Science Advisory Committee to Union Cabinets, and he's a member of the National Knowledge Commission. And he has been recognized by the government by Padma Shri and then Padma Bhushan. Okay, let's see. Okay, I think I'm almost, I don't want to keep taking more and more time. I have known him very well. It has been, I was very lucky to have known him and then continue to maintain the friendship. So with that, we'll ask Balaram to give a talk. Okay? Ram?
Ramurti, I'll share my screen.
Okay. Yeah. Okay, let's see. I had to end the show. Let's see, how do you? Oh, maybe I should have done this. Okay.
Can you see? Tell me if you can see my screen.
Not yet. I think I have to do something. Okay, now it's not. You are seeing my screen.
It's coming up. Yeah.
Oh, okay. Okay, I have to get rid of this.
Yeah, we can see.
Yeah, I can see yours.
Okay, you can see my screen. I'll make it full screen.
Yeah.
Okay. It's not full yet.
It'll become.
Okay,
Yeah.
now it's full.
Okay. Shall I start, Ramurti?
Yeah, you go ahead.
Okay, good. Well, good morning to those of you who are in America, and good evening to those of you who are in India. The task that Professor Ramurti has given me is to talk about Professor G.N. Ramachandran. And the title of my presentation could well be G.N. Ramachandran and his plot, or the story of the stereochemistry of polypeptide chain configurations, which is the title of his famous paper in the Journal of Molecular Biology. In the announcement for this lecture, Professor Ramurti had said that this would be a general lecture not related to photochemistry and photophysics. It is also not a lecture which would normally be given to an audience of organic chemists. But what I want to show you in the first few slides is much of Ramachandran's work can really be rightly classified as an extension of the principles of organic stereochemistry to the structures and conformations of very large molecules. There are two words that I would like to draw your attention to: configuration and conformation. You learned these in preliminary classes in organic chemistry. But configuration means something slightly different for people who work in polymer chemistry. And in Ramachandran's paper, he's used the term stereochemistry of polypeptide chain configuration, because he uses it in the way it was used then in polymer chemistry. But the word stereochemistry is there. And the journal in which it was published was a new journal at that time, just started in England, with John Kendrew as the editor, the Journal of Molecular Biology. So you can see this is one of the earliest kinds of papers in the Journal of Molecular Biology to use the term stereochemistry. On the extreme left of my slide is the Ramachandran plot, or the Ramachandran map as it is known. And as I go along in my lecture, I will tell you how this map actually evolved. But before I do that, I want to show you one historical slide. In 2017, in the New York Times, there was an article which said how Pasteur's artistic insight changed chemistry. And this was an article which drew attention to another commentary in Nature Chemistry on Pasteur and the art of chirality. When I read this, I thought I must share this with you, because when you think about chirality in nature, you immediately go back to Pasteur's famous lecture of 1860, which is translated into English and which I have on the top right. The lecture is not really available now. It was reprinted 3 years ago by the Alembic Club. I haven't got a copy of it with me, but I recollect from the days I read it carefully that Pasteur asks a question after his work on tartaric acid. He asks, are the atoms arranged at the corners of an irregular tetrahedron or on the spirals of a dextrogyrate helix? This is the first time, I believe, that the term helix would have been used in connection with...
Balaram, I'm
Yeah.
going to mute all and then you can turn it on. Because there are some interruptions. Yeah, okay. You just unmute.
Yeah, I've unmuted now. Can you hear me?
Yeah.
Pasteur asks this question. For an asymmetric molecule, he asks the question, are the atoms arranged at the corners of an irregular tetrahedron or on the spirals of a dextrogyrate helix? I'm going to use the word helix many times, but I think this is one of the earliest times that it was used in the literature of what was going to be the field of stereochemistry. Now I will have to stop sharing and start again because this happens whenever you do anything.
You should be able to share.
Wait a minute, I just stop share. Share again. This is the problem if...
Hmm?
Come back.
Yeah, it is not multiple share.
Now it'll move. So I just thought I'd put this up to remind you of the origins of the word helix in stereochemistry. But I was struck by Ramamurthy's reference to photophysics and photochemistry. And I just wanted to put up this article. It's a review of work which I've been aware of for some time, but this deals with the conformation of bilirubin, which is the metabolic degradation product of heme. This is the molecule which gives you the yellow color in jaundice, and it is bound to the protein serum albumin and transported away. But in order for albumin to bind bilirubin, bilirubin must undergo a photochemical EZ isomerization. This is what is used in phototherapy for neonatal jaundice, which babies are taken out into the sunlight, or better still, a fluorescent tube light is placed in the incubators. But the conformation of bilirubin is again analyzed using torsion angles. And what Ramachandran really introduced into the literature for complex molecules was to use torsion angle space as the conformational space of the molecule. And one last reference to tell you about the importance of this work even in chemistry. This is a paper which has appeared just very recently in Nature Chemistry. The title is interesting. It says, illuminating the dark conformational space of macrocycles using dominant rotors. And then I've simply made what today is popular as a graphical abstract from this paper. But I'll read you just one sentence. It says, the observation of unusual conformations of 16 to 22 membered rings has been made possible by controlling their interconversion using dominant rotors, which represent tunable atropisomeric constituents with relatively high rotational barriers. This is a description of the work in this paper in very complex terms by using the word atropisomeric. But it turns out that much of what is written in this paper is understood in terms of Ramachandran plots and the excursions that polypeptides make in Ramachandran space, which is why the right-hand side of the slide shows you the two Ramachandran plots, which are actually shown there. The relationship to organic stereochemistry is clearly made by looking at the picture on the left. You could see the cyclohexanes illustrated and down below peptides illustrated. But let's go back to Ramachandran's paper. This is what the first page of the paper looks like. I'll come back to this paper later. But effectively, Ramachandran looked at a polypeptide as a system of linked peptide units. Peptides are planar and they're connected by a tetrahedral carbon atom. So effectively, the geometrical problem was to analyze all the possible orientations of planes which are connected at a tetrahedral hinge. Ramachandran's work is applied to protein conformation. And you might ask, are proteins important? To biochemists, to cell biologists, to molecular biologists, proteins are very important. On the other hand, sometimes to the general public, proteins are not very important except as a source of nutrition. But the coronavirus has changed all of this. Everybody now knows the picture of the coronavirus and those spiky projections on the surface of the virus. Those are proteins. And a virus itself was defined many years ago by the British immunologist Peter Medawar, where he called a virus a piece of bad news wrapped up in protein. And that's what a virus is. So the outer cover is largely protein. And I'll come back to this toward the very, very end of my talk. But to just show you what those proteins look like on the surface of the coronavirus, this is what they look like. This is the so-called spike protein, the protein to which all the antibodies will bind, the antibodies that you generate when you take a vaccination. But look at the picture. What does the picture show you? The arrow that I have on the top left corner of the slide indicates the part of the protein which is most important and in which everybody is interested. That is the receptor binding domain. If you can see, that is represented by those green arrows. Those are secondary structure elements, beta strands. If you look at the lower part of the structure, you will see spiral structures. And this is just a long polymeric molecule in which the folding of the polypeptide chain is represented in this fashion to indicate what we now call secondary structure elements. So these are important whenever you're looking at protein structures. You know, proteins themselves go back a long way. Part of the difficulty in understanding enzymes, for instance, was because of the divide between organic chemistry and the emerging field of biochemistry. Richard Pilstarter, for a very long time, held the view that you cannot have a macromolecule. What you can have are colloidal substances. And he thought that proteins were colloidal substances. It was only after Sumner and Northrop crystallized proteins that it became clear that proteins were just large organic molecules. That's what they were. They were large biopolymers. But I show you a little bit from the Pauling blog. You will find that in 1936, on the side of a book that he got from one of the founding fathers of immunology, Karl Landsteiner, with whom Pauling was collaborating on the specificity of antibodies, he's made notes on the margin. What he says in the margin is, I think the protein must contain side chains with active groups. A remarkable statement if you go back to 1936, because at that time, nobody knew what the constitution of proteins really were. We've moved a long way since then. All atom structure of proteins can be determined by crystallography. It will look like the picture that I've shown you on the top left of the slide. A completely meaningless connection of atoms. But proteins become more beautiful, more manageable objects when you represent only the backbone. And you represent the backbone using the arrows to represent secondary structure elements, strands, and the spirals to represent helices. So effectively, proteins are made up really of helices and strands connected by irregular elements of structure. Our view of helices became clear only when this book appeared in 1969. This is a book by Richard Dickerson, which was on proteins, and it was illustrated by one of the most important illustrators in science, Irving Geis. There were no computers at that time, so Geis drew these drawings by hand. And you can see in the Irving Geis drawings, you can see what a helix looks like. You can see the amide planes connected by the tetrahedral carbon atoms. And as a helix, you can see that the planes sort of move around an axis, and eventually hydrogen bonds hold the helix together. Today, if one is teaching a class, and I've taken this from a transparency which I used to use many years ago, this is how you would illustrate the secondary structures in protein. This is what the Ramachandran plot brought vividly into everybody's thinking in an analytical fashion. What are helices, what are sheets, and how do you define them? The Ramachandran plot effectively defines the limits allowed of polypeptide stereochemistry. Along the two axes of the Ramachandran plot, the x-axis, which is phi, and a y-axis, which is denoted by the letter psi, you have torsion angles. A torsion angle is what I have represented at the bottom here. If you have two atoms which are connected together, you have only one bond length. If you have three atoms which are connected together, you have two bond lengths and a bond angle. But if you have four atoms connected to one another, you have the dihedral or the torsion angle. Atoms 1 and 4 can be outside the plane of the board or the screen. And that is the rotation that you indicate above the 2-3 bond. It's the torsion above the 2-3 bond. In order to define a torsion angle, you need the x, y, z of coordinates of 1, 2, 3, and 4 in three-dimensional space. And then you can actually define the angle. But in a polymer, you will have many such angles. For proteins, you have two such angles for every amino acid residue in the chain. So you can imagine if you have 100 residue protein, you would have two angles around each amino acid. You would have a huge number of torsion angles. Ramachandran map consists of islands of allowed structure, and much of conformational space is disallowed. And I'm going to show you how this happened. Most importantly, it allows the transformation of a three-dimensional problem into two dimensions where we can now think analytically. How has the Ramachandran map been viewed in the literature afterwards? I've taken two fairly recent papers, 2011, one by George Rose and the next one by Lynn Reagan. I've taken a sentence from this. It says, no general biochemistry textbook is complete without a phi-psi map of the alanine dipeptide. This iconic plot is a compact representation of a profound organizing idea, one that ranks among the fundamentals of structural biochemistry. And in Lynn Reagan's paper in the same year, it says that the Ramachandran plot is an iconic image of modern biochemistry. And then she says at the end of her paper that in summary, we have shown that the distribution of backbone dihedral angles observed in proteins of well-known structure is explained by Ramachandran and co-workers' original analysis. And then she says, we find no need to invoke additional interactions to explain the backbone conformations of proteins. This means that the Ramachandran map is still completely valid in all discussions of protein structure. So here then is a picture of Ramachandran, taken a long time ago, standing next to a board in which he's drawn two helices in two dimensions, the alpha Linus Pauling's alpha helix on the left and his own triple helix on the right. This is Ramachandran again, a later picture, and the Ramachandran map now, of course, colored, and with the different regions of secondary structures now marked upon them. I joined Ramachandran's department in 1973. He left it in 1978-79. But I stayed in touch with him until he died in 2002. But in 2003, shortly after he passed away, the proceedings of the National Academy of Sciences of the United States celebrated 50 years of Linus Pauling's famous papers on protein structure. And in an article which celebrated this Pauling event, David Eisenberg had this paragraph. He said that today the Ramachandran diagram is taught in all classes on protein structure and is featured in every textbook to give insight into the forces that determine the structures of protein. But there is nothing in this diagram beyond what Pauling and Corey knew well. They built models of their proposed structures that embodied all features of the Ramachandran diagram. Apparently, they understood the principle so well that they felt no need to explain them by a diagram of the sort. As I go along, you will find that almost everything that Eisenberg has written in this paragraph is probably overstated, incorrect, and unfortunately archived now in the literature. Let's go back to history of polypeptide chain stereochemistry. In Chemical Reviews, Maurice Huggins published a paper in 1943 where he wrote the first hydrogen-bonded structures of polypeptide. You can see them. What is today called a projection of a 2,7 chain or a projection of a 3 chain. A few years later, in this paper in 1950, in the Proceedings of the Royal Society, Bragg, Kendrew, and Perroux drew these structures of polypeptide. I draw your attention to this paper because, as far as I know, this is the only paper written by 3 authors, all of whom received Nobel Prizes. One of them before the paper was written, the other 2 after this paper was written, in which every structure in this paper is actually wrong. And this is because at that time, they did not make use of the knowledge that Pauling had, that the amide unit is planar. That is, the peptide unit is a planar system. This is Pauling in Madras in 1967, when Ramachandran held a symposium showing a picture of what he had discovered, the alpha helical structure of polypeptide. That happened in the early 1950s. But in Madras, Ramachandran moved to Madras in 1951. At a very young age, he was barely 30, he was already professor and the head of the Department of Physics. He was trained at the Indian Institute of Science under C.V. Raman, under whom he got his PhD, working in the area of optics. He was later on the faculty of the Indian Institute of Science as an assistant professor, and it was there that he began his work in crystallography. So when he left Bangalore to go to Madras, Ramachandran was an expert in optics and an expert and beginning to be an expert in crystallography. He needed a problem to work on, and at that time, J.D. Bernal, who visited Madras, suggested that the structure of collagen was unknown. This was the time when the structures of proteins were being studied. Collagen is the most abundant protein in animals, and therefore collagen is a very important molecule. The Central Leather Research Institute was next door to Madras University, and there Dr. Naidama helped Ramachandran obtain a sample of collagen, and he obtained the fiber diffraction photograph, which I show you on the slide. This is the early 1950s in Madras, and Ramachandran had one co-worker, Gopinath Karta, who was a remarkable man, who was Ramachandran's postdoc. Postdocs were rare in those days. He joined Ramachandran in Bangalore, moved with him to Madras. Together, Ramachandran and Karta began to study collagen by fiber diffraction. Collagen is a large fibrous protein. It's the major component of bone, tendon, cartilage, and teeth. It's the most abundant mammalian protein. It has a very unusual amino acid composition. The only other thing that you need to know is proteins are polymers formed by linking monomeric amino acids together. The unusual amino acid composition of collagen gives you a clue. 1/3 of collagen is the smallest amino acid, which is glycine. And the bulk of the other amino acids are proline. And proline is sometimes post-translationally modified to make hydroxyproline. So the sequence is really of a tripeptide, glycine, proline, proline, repeated many, many times to give a long fibrous molecule. Ramachandran and Karta interpreted the fiber diffraction pattern. They also knew that 1/3 of the amino acids had to be very small. And therefore, in order to maintain the threefold symmetry that they get, they began to look at triple helical structures. If you have 3 strands which are wound around the common axis, you can now get 10 threefold symmetry for the structure. Now, what must be inside if you're winding strands? It must be something small so that it can be accommodated, and therefore the amino acid constraint of glycine. Eventually, they produced this model. This is the triple-stranded structure of collagen, built many years later with metal peptide units, screws, Kenru-type models. This model it was built in Bangalore, and you will see this model now on display just as you enter the auditorium of the Central Leather Research Institute in Chennai. Madras is now Chennai, and the auditorium is called the Triple Helix Auditorium. It is probably the only major building which is not named after a politician. Therefore, I think it is named after a scientific structure. This is a great tribute, I think, to Ramachandran in many ways. These are the famous papers on I want you to only look at the dates. Ramachandran in 56, Ramachandran and Karta in 55, and the first paper, Ramachandran and Karta in 54. Francis Crick of DNA fame and Alex Rich, 1955 in Nature. So you can see that different people were trying to get the structure of collagen. Those of you who have read the double helix, where Watson describes in racy terms the race for the double helix, will recognize some of the characters in the collagen story. Francis Crick is one, and Randall at King's College in London is the other. But no story has been written about this as yet. In August of 1954, Ramachandran sent his paper with Karta to Pauling, who was also working on the structure of collagen. And Pauling was non-committal. He said he'd read it with interest, and he says he's not able to reach a definite conclusion about the probability that the structure is correct. But he says it seems to be unsatisfactory in some respects, in particular, the bending of the hydrogen bonds. Remember, these were early days of hydrogen bonds, and that was Pauling's comment. This is physics in Madras. It had not yet become biophysics and crystallography at that time. And this picture, taken in 1955, told you what the people looked like. Also tells you something about what India was like. Today, if you come back and take a picture in front of any of our major departments, you will find people colorfully dressed. You will find pictures in color. And this is what we were then. There's Ramachandran, GNR, and there's Karta. I've named some of the others who are present in this. R. Srinivasan, who later became head of the department and who worked with Ramachandran on crystallography. Shashi Shekharan, who worked with Ramachandran on biophysics. I'll show you some correspondence. For all of this, I have to acknowledge Professor Yatindra, who used to be at the Madras University's biophysics department and who had managed to get these letters and sort of scan them and make them available. This is a letter dated 1st November 1955 by Francis Crick. And this is a long letter. It is 3 pages long. For those of you who are biologists here and who venerate the Watson-Crick double helix, you must remember that the Watson-Crick double helix paper appeared in Nature in 1953. Ramachandran's collagen paper, which is the triple helix, appeared in 1954. And Crick has written 3 pages of a letter. It's a detailed technical letter. But at the end of it, he says, we should be very glad to have your comments on these remarks. We're holding a seminar on collagen here on 14th December, and we should like to present your latest ideas on the problem. May we do this? Or perhaps you would prefer Dr. Karta to do it. By this time, Dr. Karta had moved to Cambridge, where he spent a short time before he moved on to the United States. Here's another letter. This letter is written by Karta to Ramachandran on the 4th of December, 1955. And I've just marked one part of the letter with an arrow, and I have transcribed it at the bottom. You can see that people, how they used to write letters. They would come by aerograms after many days. This was the case even when I was abroad in the 1970s. And we would fill the aerogram, and therefore Karta's handwriting is correspondingly small and difficult to read. He says here, Dr. FHCC, that is Francis Crick in Cambridge, was telling me, then I left the rest out. I do not know what he meant, but the general idea seems to be that the publication of our paper was purposely delayed by months for them. By them, he means Randall at King's College, London, to work up our idea of seeing the manuscript. Elsewhere in this letter, he says that Francis Crick is unhappy that Ramachandran's paper may appear before his, the second paper that is, and suggesting that the two papers be combined together so that the Chennai and the Cambridge structures could be published together. But Karta was a remarkable man. 15th of December 1955, just after the symposium was over, he reported back to Ramachandran. And then he says here, I marked only this. I'm also worried about the large number of free NH groups. And from that point, I would prefer a 2-hydrogen bonded system, even though that involves bad contacts and a bit of strain to make R2 a proline. Ramachandran and Karta understood the principles of organic stereochemistry, the strain in proline. They understood bad contacts, which was being talked about. And these are what eventually led to the Ramachandran plot. Crick's criticism of Ramachandran's original structure led to a great deal of difficulty for Ramachandran. In those days, Ramachandran was a young man. Remember, he was born in 1922. So by the time his collagen papers had appeared, he would have been 34 years old. He was a young professor in an Indian university, a traditional university, and he would have undoubtedly been the subject of great jealousy. And therefore, the environment would have treated him and talked about all this quite a bit. But Ramachandran on collagen many years later pays the tribute to Pauling, and then he says that the helix era had begun in 1951-53. He says, I mention all these because I was encouraged to enter this field by reading Pauling's papers and how he began to do crystallography. He acknowledges J.D. Bernal here. And I show this to you because in 1988, when Ramachandran wrote for the International Journal of Peptide and Protein Research, then edited by the famous endocrinologist Chow Lee at the University of California in San Francisco, I carried the manuscript of this paper to deliver to C.H. Lee. And this paper, many years after all of this had happened, Ramachandran wrote about collagen. There's the alpha helix, there's the DNA double helix in 53, and there's the Ramachandran collagen triple helix in 54. So Ramachandran was extraordinarily contemporary. And more than that, if you ask, which of these three structures was the most difficult to determine, it was the collagen structure. The complete constraint of hydrogen bonding was not there, as in the case of the alpha helix and the DNA double helix. Collagen was, in fact, a triple-stranded structure with relatively little contributions from the hydrogen bonds, and one needed to interpret very little fiber diffraction data, which was then available. In 1967, Pauling came to Madras and he wrote this. He says that his own structure, which proposed in 1951, was not right. And then he said that in 53, he said that he and R.B. Corey should be allowed to have two guesses on collagen. And he said that we were determined that our second guess would be right. He proposed what is now known as a stochastic method of predicting structures. Then he says they never got the chance to make their second guess because Ramachandran and Karta found the structure of collagen. This is his presidential address at Madras. Crick also reflected on DNA and collagen years later in the book that he wrote. He says that the discovery of the DNA helix, the path to it, scientifically speaking, was fairly commonplace. What was important was not the way it was discovered, but the object discovered, the structure of DNA. And then he talks about collagen, and he says it's a complicated molecule, and he said it had all the elements surrounding the double helix. The characters confused and false solutions just as misleading. Competition and friendliness also played a part in the story. I don't think Ramachandran felt very much of that friendliness several thousand miles away in Madras. Yet nobody has written even one book about the race for the triple helix. It's surely because in a very real sense, collagen is not as important a molecule as DNA. Then he goes on to add, to some extent, it depends on what you consider important. He said, before Alex Rich and I worked on collagen, we tended to be rather patronizing about it. After all, we said there's no collagen in plants. In 55, after we got interested in the molecule, and Ramachandran's first paper appeared in 54, he says, do you realize that 1/3 of all the protein in your body is collagen? Then he says, it's the molecule which has the gamma and not the scientists. But it is the structure of collagen, and it's the story of collagen, and it is the criticism that Crick and Rich made of collagen that led Ramachandran to his plot. Because what he said was that Ramachandran's collagen structure is stereochemically disallowed. That means the atoms come too close to one another, and since atoms are rigid bodies, they cannot come closer than the sum of their van der Waals radii, and therefore that is disallowed. Except that the very definition of the kind of van der Waals radii that you must use was not quite well known in the literature at that time. In 2012, a single crystal structure of a large peptide corresponding to a collagen sequence was determined, and then the controversy is completely resolved. It turns out that the average molecular structure of collagen does not correspond to the prevailing Rich and Crick model, but corresponds instead to a triple-stranded 7-2 helical model. This is the kind of model which Ramachandran and his group were in fact working on. But let me come back to the Ramachandran plot, because that is what made Ramachandran really immortal in the literature of biochemistry. In the Dickerson-Geist book of 1969, this plot appeared. Ramachandran's paper appeared in 1963. He wrote a very famous review with Shashishekaran in Advances in Protein Chemistry in 1968. By that time, the plot was going to be changed in the way it looks to its modern depiction. But here is the Ramachandran plot in Dickerson's book. And Dickerson did not now call it a 5-side plot. He called it a Ramachandran 5-side plot with contours of equal length. Now, Dickerson named it the Ramachandran plot. Afterwards, this is the Ramachandran plot in the scientific literature. But Dickerson did not like the way the plot looked eventually. And this is because the IUB Commission on Biochemical Nomenclature was trying to make the plot achieve consistency with the usage of organic chemists. Dickerson felt that this would make the literature virtually unreadable. Today, it turns out that the literature of organic chemistry and the literature of biochemistry have no difficulty as long as the torsion angles have the right kind of signs attached to them. But Dickerson says something interesting. According to traditional sources, there are 2 types of sin: sins of omission and sins of commission. This is most definitely a sin of commission. But the IUB Commission on Biochemical Nomenclature prevailed. And this is what the Ramachandran plot looks like. This is what happened. In 1962, the plot appeared like this in the proceedings of a symposium at the Central Leather Research Institute in Madras, which took place in 1960, but was published in a book in 1962. This is what it looked, and eventually, this is what it looked like in the Journal of Molecular Biology paper in 1963. You can see it's sort of inverted because the conventions of nomenclature are different. And this is the second figure in that paper. But I'll come back to that. But how does one calculate dihedral angles? If you have coordinates of 4 atoms, it's a fairly complex arithmetic job or algebraic job to compute the dihedral angle. It's easy enough to do it for 2 points, slightly more difficult for 3 points, somewhat more difficult for 4 points. But if you're going to do this for 4 points and you're going to change the angle by 10 degrees or 5 degrees each time and cover all of space, This is a lot of calculation. This was done by Ramachandran's PhD student, C. Ramakrishnan, who later became my senior colleague in the molecular biophysics unit at the Indian Institute of Science. Ramakrishnan was armed only with two implements, Clark's tables, which gave the logarithms and the antilogs, trigonometric functions, and this calculator, which had no trigonometric function. So he would calculate. He calculated days, he calculated nights, he calculated weeks, and he calculated months. And this is, of course, Ramakrishnan many years later, teaching students at the Indian Institute of Science, Education and Research in Pune when he was on a visit there, how to do these calculations. Nobody will do them nowadays. There are programs which will do them, and there are sites to which you can submit your coordinates. But Ramakrishnan remained till his very last day someone who could do it himself by hand. So what Ramakrishnan did really was to change the phi-psi values and look for bad interatomic contacts. He did it for all of conformational space and put it onto this two-dimensional map. So immediately you can see which regions of conformational space are available and which regions are disallowed. This square that you see represents all conformational space. Out of all of conformational space which is available, only about a third of it is stereochemically allowed. But I want to show you something, and by now you would have had enough time to look at the slide. If you go to the Journal of Molecular Biology in 1963, and if you look at the figures in the paper carefully, you will find that the legends to the two figures are interchanged while printing. They've never been corrected. Many years later, when I was producing a special issue of the journal to which Professor Ramamurthy alluded, Current Science, and we were doing a special issue on Ramachandran, I wanted to reprint this, and I got permission from the Journal of Molecular Biology to reprint the paper. But when we were reprinting the paper, I realized that the legends had been interchanged. I then asked my colleague Ramakrishnan, and Ramakrishnan passed away only a couple of years ago. I asked Ramakrishnan then, shall I make this correction? But Ramakrishnan told me, no, you cannot make this correction. Professor, and this is what he would call here now, Professor is still there, and he will get angry. This was 27 years after Ramachandran's paper was published. But what this does tell you is nobody ever reads the literature, but what they read is the other secondary sources. And from the secondary sources where things have then been corrected, everyone probably then gets it. The famous Ramachandran paper appeared in 1963. And you can now ask the question, how many protein crystal structures were available at that time from which he could get phi-psi values for every single amino acid and put them onto his map to see whether experimentally his map could be validated. It turned out there was only one structure at that time at that kind of resolution. That was myoglobin, done by John Kendall. Hemoglobin at that time was still at a resolution that some of these numbers could not be taken terribly seriously. To emphasize this historical point, I show you this picture from a 1965 lecture by David Phillips at the Royal Institution. The paper was published in Nature in 1965. This is the first enzyme structure, the structure of N-egg white lysozyme. This is David Phillips lecturing to an audience at the Royal Institution, where you can see at the bottom left of the photograph a complex molecular model of lysozyme. I show you a modern representation on the extreme left of the slide. But on the extreme right of the table, where he is demonstrating the structure, you can see a chain. And what he says is that if you took the lysozyme sequence and took it as a string, it would be 32 feet long and it would reach the ceiling of the Royal Institution. But you can see that the British scientists at that time worked in London, they worked in Cambridge, they worked in Oxford. They worked in environments which were completely different from the environment that Ramachandran worked in the early 1950s in Madras. The 1968 review by Ramachandran and Shashishekaran is very, very important. It is, in fact, a Bible for everybody who enters the field of polypeptide conformation. It is a classic article. But Ramachandran, even then, was a very strict man. In his communication with John Edsall, who was then the editor of Advances in Protein Chemistry and a very famous protein biophysical chemist at Harvard, at the Harvard Medical School, Edsall has written this note. He says, by the author's request, the publishers have left certain matters of usage and spelling in the form in which they wrote them, because Ramachandran would not move from the position that he took once he had written something. The evolution of the Ramachandran map, as you can see it, is like this. What it looked like in 1960, what it looked like in 63, in 68, and today if you take a Google image. I would like to address the criticism that David Eisenberg made of the Ramachandran map in 2003. This article appeared in Angavantikami, and chemists would like this. It's called Pauling's Left-Handed Helix, and it is by Jack Dunnett, who was a postdoc with Pauling. And he says, a few months after the publication of Pauling structures, Morris Higgins noted that for amino acids with a correct absolute configuration, then he adds, he was already aware of the barefoot result. A left-handed helix would lead to a C-beta to oxygen distance of only 2.64 Armstrong. He concluded that levopolypeptides form right-handed spirals and dextropolypeptides left-handed spirals, whichever of these two types of structures is correct. The protein chain has to be a levopolypeptide because it has L amino acids. But Pauling, if he was aware of this, we do not know, but every coordinate that Pauling gave in his famous papers is for polyglycine, which is achiral. And therefore, Pauling sidestepped the issue of these short contacts, which led to the Ramachandran map. But today, if you had the Ramachandran map, you would automatically be able to tell whether the helices should be left-handed or right-handed. Today, if you solve a protein crystal structure, and I use the structure, a famous structure of Rubisco, a very widespread protein in nature, which was incorrectly determined. And this is what the Ramachandran map looked like in 1988. If you correct the model, this is what the Ramachandran map looks like. All the black dots now, which are individual residue conformations, have moved in to within the allowed regions. Everything which lies outside the allowed regions are glycine residues. So today, crystallographers must use the Ramachandran map as the first stage filter to determine the correctness of the structural model. A famous crystallographer, Brian Matthews, actually wrote a tribute to Ramachandran by showing these pictures from one of his own structures. At 2.8 angstrom, what would the map look like? What would the map look like when the resolution was improved? Today, after 50 years and more, this is what the Ramachandran map looks like. Hundreds of thousands of crystal structure points are now available. They give you an experimental Ramachandran map. You can now plot the Ramachandran map in much more colorful ways. You can make them three-dimensional. You can look at the intensities of the points that you get at any region in Ramachandran space, and you can do global analyses of protein structure. Today, we live in the age of scientometrics. Everybody counts citations and the various kinds of indices which define both journals and scientists and institutions. How would we view Ramachandran in the age of scientometrics? I show you, and this slide I made a couple of years ago. You can see what it looks like. Ramachandran's articles in the Advances in Protein Chemistry and the Journal of Molecular Biology are shown there. you can see that these articles have increased in the number of citations that they get as time has progressed. And in the 21st century, they are actually getting far more citations than they get in the 20th century. And occasionally you will find, and if you're a spectroscopist, as I was, you might sometimes try to deconvolute the curve that you get here to see whether there are two peaks. And you might ask, what is it in science that has caused the sudden interest once again in the Ramachandran plot? To do that, you will find that at one point, when synchrotron radiation came into being and crystal structures of proteins began to be determined with greater rapidity, more structures appeared in which the Ramachandran paper was quoted. Another field also developed, which won a Nobel Prize a few years ago. That is the field of molecular dynamics as applied to biological molecules. When molecular dynamics appeared, it turns out that all the dynamics that you do is done in torsion angle space. And analysis is done in torsion angle space. So every molecular dynamics paper which analyzes polypeptide trajectories now uses the Ramachandran kind of analysis. You can see that a paper by Ramakrishnan and Ramachandran in 1965 has been cited very heavily in the year 2000. And this is because this is again another landmark paper. And right at the bottom, I show you a very highly cited paper. Ramachandran's name is not there on this paper. It's from the Madras group. It is the paper which introduced the beta turns into the literature, a very important paper, a structural feature in protein. There's a single author, Venkatachalam, who was Ramachandran's PhD student. And Ramachandran was angry at the time when this paper was written, apparently, and Ramakrishnan told me that he struck his name off the paper. And this, of course, is another major contribution which came from the Madras group. How has Ramachandran been viewed in the years since? In India, Ramachandran retired, spent long years in retirement, and died in 2002. He was widely respected, widely feared, and at the same time, generally ignored. And both by government and by all the bodies in India which could have honored him. When the Indian Institute of Science celebrated its centenary in the year 2008-2009, I had the privilege of serving as the institution's custodian. And therefore, I had the privilege of designing the stamps which would be released on that occasion by the Department of Posts. The two stamps that were made are the ones that I show you on the right. The top stamp shows you the iconic main building of the Institute. The Department of Posts told me at that time that you cannot put the picture of a single individual on an Indian stamp. I suspect you can do that only if the person is a politician, otherwise you cannot do it. But they could not argue when I said, I won't put the picture of one person, but I will put the picture of many people. They said, how many? I said, I'll fit them into the stamp. And so here you see many of the people who have been associated with the history of the Indian Institute of Science. Jamshed Tata, of course, right at the beginning, was the founder of the Institute. Raman as its first Indian director to his left. Satish Dhawan, the longest serving director of the Indian Institute of Science, and in many ways the architect of India's space program. And then next to Satish Dhawan on the extreme left, you can see the triple helix of collagen and Ramachandran, because Ramachandran is probably one of the most distinguished alumnus of the Institute. I will not tell you who the other people are. Students can take it as a quiz. But Ramachandran was always not treated very well. In the early 1980s, he'd gone to America in 77 and 78. And his income tax assessment, I searched the internet to find what references I can get on Ramachandran, and I found this. He had a case where the income tax department wanted to take back the money that he had been paid for his visit in the United States. He'd obviously given some lectures at the National Institutes of Health and in other places, and he'd received some honoraria. And he'd also received some expenses for living in the United States for the year that he spent. But the income tax department wanted to tax these, and Ramachandran appealed. Eventually, he won the case. When in appeals, the judge held that the amount received the SSC was not in the nature of salary, but in the nature of scholarship. Ramachandran contributed in many ways to scientific scholarship, and it is sort of appropriate that he should then also be rewarded with a monetary scholarship of a very meager amount that you see. This is a picture of Ramachandran at his prime. Next to him is the Vice Chancellor who protected him at Madras University during his years there, Lakshman Swami Mudaliar. Next to him is Linus Pauling, and next to him, Pauling, is Dorothy Hodgkin. The two of the most famous figures in modern chemistry, Pauling and Hodgkin, in Madras in 1967 to attend Ramachandran's symposium. I searched the internet to see if I could find anything about Pauling in Madras in 1967. And I found one letter in the Pauling archives. Unfortunately, the letter is not from Pauling, but from Pauling's wife, Ava Pauling. And she's written to her son, Peter Pauling. She says, we've been having a busy time. It's interesting too. I really can't see any future for India unless they change drastically. It is sad. The women do a lot here. I think India has changed very much. Mrs. Pauling was probably wrong in her assessments of Madras at that time. The scientific assessments of Madras at that time were extraordinarily good. And much of Ramachandran's work now has passed into the biochemical literature and is used for analyzing even current day problems. I would like really, in conclusion, to show you this slide. This is the sort of slide which everybody would be interested in today. It addresses the SARS-CoV-2 problem. The protein structure that is on the top of the slide is the spike protein structure. At the bottom of the slide is the cellular angiotensin converting enzyme receptor on your lung cells, which allows the virus to get into the cells. These two proteins interact. Today, these are the targets for therapeutic and vaccine design. How does one address therapeutic design? You have to address the protein structure, the molecules which bind to the protein, the molecules which interfere with protein-protein interactions. How do you design vaccines? You have to design antigenic molecules, proteins, which will eventually give rise to antibodies, which will neutralize this kind of interaction. So all those who sit now happily vaccinated against the coronavirus pandemic should do well to realize that proteins are central to almost everything that we're talking about today. The solutions to these problems are not evident. The scientific solutions to these problems should have come when the SARS-CoV-1 episode took place. There was enough scientific literature at that time, but funding was then not available. People did not proceed very much. The kind of activity which you have seen over the last 1 year was not there. So there's a lesson here. There's a lesson that basic science, fundamental science, and in the case of Ramachandran, fundamental stereochemistry, is critically important in understanding many of the problems that confront us and which will confront us in the future also. In conclusion, I will show you Ramachandran's picture once again and also provide you with a reference to a documentary that has just been produced by the Indian Institute of Science, Education and Research. It's available on YouTube and it has been correctly titled The Trials and Triumphs of D.N. Ramachandran. The most unfortunate part of it is, I think, Ramachandran passed into history without quite recognizing how widely his work was used in the literature. Lastly, I'd like to thank the two institutions which have provided me a home for all the years, my professional years, and in my retirement years. The Indian Institute of Science on the top left and the National Center for Biological Sciences at the bottom right. In both these institutions, provide you the luxury of doing whatever you want, reading whatever you want, writing whatever you want, and most importantly, thinking about whatever you want. Thank you very much.
All right, thank you. Dr. Balaram,
Yeah.
is, would you be willing to answer a few questions?
Sure. If anybody has any.
Yeah, let's find out. Okay. Anybody questions? Okay, let's see. Yes. Anybody with questions?
So I have a question. I'm Govinda Rao. I also graduated from IIC Bangalore. One question already bothered, right? Like politicians not recognizing Ramachandran is a country ignoring is okay, but why did the peers did not recognize him, in India while he was living? I was in the campus of IIC from 1980 to 1987. He was still there, right? But we didn't give him the recognition required. What happened?
Ramachandran was, I would urge you to look at the documentary, hear it. Ramachandran's son will speak in that documentary. Ramachandran, in the years immediately after college, had more than once episodes of breakdown, and how much of it was caused by the stress of the collagen controversy, how much of it was organic, we do not know. But he did have this problem. But at the same time, he was, I think, in many ways, an idiosyncratic person. He could get very angry, and he could get very angry very quickly. And therefore, I think people generally tended to avoid him in his later years. And once he retired and went away, he was in the public view. He almost disappeared from public view.
Yep. Thank you. That is unfortunate, but I know that he did not get Nobel Prize, or he was not Padma Vibhushan or anything like that. But as you said, Professor Balaram, his name is immortalized, right? There is no structural paper which comes out without his name, his plot, right? So so he is recognized in many, in a higher sense, right?
He could have been much better recognized in India before he passed away. Unfortunately, it did not happen.
Can I ask a question?
Yeah.
Sure.
Yeah, this is Jyotishman Dasgupta. Professor Balaram, thank you very much for this very nice talk. And as usual, I love your lectures and the scholarship in them. I just wanted to ask a question with regards to the timeline of the work that GNR had done. And this was from 1954, 1955, 1956, all those papers that came out in Nature. Do you have some sort of a semblance of how much time did it take for him to sort of present that model, and that 1 year gap between all those papers? I'm sure those are just basically maybe things have worked out previously, and he was trying to put things out in succession. But do you have any semblance of idea how long did he work on these models before he published the data?
At that time, he worked intensely on collagen, and I suspect that it didn't take them very long, because both Ramachandran and Karta, whom I knew in later years, were remarkable people. Karta was a very, very modest man, a very quiet man. He solved the first enzyme structure coming from North America, the structure of ribonuclease, which he did with David Harker. And I saw Karta's letters to Ramachandran only now, when Yatindra sent them to me a few months ago. And when I read those letters, I come to the conclusion that they knew a great deal which we don't give them at that time itself.
Right.
Fortunately, I knew Karta in the 1980s, and I did not know what I know today, so I could not ask him.
Sure.
If I
sure,
had
sure.
known, I could have asked him.
Yes. Yes. Sure.
But I did not know what I know today. That's the problem.
Yeah. Okay. Thank you.
Balaram, can I ask a question?
Sure.
Can you hear me, please?
Yes, of course.
Yeah, thank you. It's always, as you know, really wonderful to listen to you. Whether you were lecturing us in 1980s or now, you haven't changed much. Thank you. A couple of things I want to bring in here. One thing is, people in science still put ego and person in the front of scientific achievements of humanity. That is much more important to see where we were and where we are as humanity than whether or not an individual person achieved something or not. I think in that second part, thinking that we are above the science brings all these politics and other things into work, which we should ignore and go beyond. Working in space sciences, I understand that it is generational effort. Now, this is my comment. So that would solve a lot of problems. My question is, we have come really far in understanding the atomic, molecular, and macrostructural aspects of life. But we still have not understood how lifeless has transformed into life. And that is one of the biggest challenges we have. Do you have any ideas and suggestions how we can go about doing that?
You see, the question you're asking for anybody who works with NASA and places like this is, would there be life on another planet? If I were to rephrase the question, what you would be asking is, can biochemistry as we know it evolve on another planet?
Or even on Earth? Or even on Earth, how did it evolve?
We don't know how. So to do that, we must know how biochemistry evolved on Earth. So that is what you call a vital question. And this is the title of a book written by a man who thinks about this a lot, a biochemist called Nick Lane. And I would urge you to buy this book and read it. It's quite remarkable. The evolution of biochemistry has a lot of people now making progress, and some remarkable progress is being made. I think maybe 50 years from now, we will know far more than we do.
Balaram, this is Ganesh here from ISA Tirupati. Excellent talk, as always. You mentioned that nobody has written a book on the race to collagen triple helix. I think you are the one who is most eminently suitable for writing this book with all your scholastic, analytical ability, depth of knowledge. It would be a great service if you could just, document and really put the facts in its historic and analytical perspective.
Here, I would make a comment, Ganesh. One of the real problems in India in documenting anything is the absence of records. And the fact that sometimes records are not read, but jealously guarded and not even made accessible. And we have not had a sense of history. And therefore, it makes any writing, unless you get documentary evidence or something that's based on hearsay, if all the people who were participants were alive, you could question them and get their versions and put together a story. Otherwise, it's difficult to do something that is authentic. But see, one would need to look at the Crick correspondence, one would need to look at the papers everywhere before one really puts it down.
Yes, I do agree. But I think with available records, whatever is a reasonable account would be a very nice thing for historic purpose. And only you can do that.
It should be the Indian view.
Dr. Balaram, make a comment. This is Srivatsan. Thank you very much.
Hi, how are you?
I mean, as usual, it's a wonderful talk. One of the things Murthy asked, and you gave a wonderful suggestion. Biochemistry, we don't know where it is going. I saw in NASA's, it seems, Avyars more than 4,000 years ago, or 2,000 before Christ, maybe. She says, "Kattadu kaivan alavu, kalladadu ulagalavu." So it's going to be very difficult. So we may be probably talking about Ramachandran's plot probably 200 years from now still, before they really know whether the life exists in Mars or not.
Okay, yeah, that's a comment. I don't have any comment on your comment.
Hello, this is Ram Krishnamurthy from Scripps at La Jolla, California.
Oh, yes.
It's a pleasure, first of all, to listen to this lecture. It's mind blowing to think how much has been achieved with whatever little they had in those days, and we have even more sophisticated instruments, and we are all struggling to understand even more. It's always inspirational to listen to these kind of lectures to find out how the early pioneers, with very little what they had and the great intellect they must have had to solve these problems. It came from within with so many things. One beautiful example you showed was the small merchant computer that they had to solve this. It's simply amazing how much they did with what little they had. That was, just a comment and an observation of this beautiful lecture. The second point that I wanted to make was with what Murthy had started this conversation of asking the questions of, how did life come about from the lifeless? It's an age-old question, which has fascinated people, and the answers range from, anywhere from philosophical to religious to scientific, spans the whole spectrum, and we all struggle with this. And it is one that has gripped my imagination right from the lecture that I heard from Albert Eschenmoser, when he said, why not hexose nucleic acids? What is so special about ribose that is part of the RNA? Whereas, you talk about the ubiquitous sugars that are present, it is glucose. Why not a glucose nucleic acid? And so that set off in my mind, and I have been, in this field for the last close to 30 years now, and it's still a struggle. It's a struggle to understand, because first of all, there is no good definition of life, because life is it's a process. It's not defined by a single molecule or even by a single interaction between two different molecules. You can have protein, peptide, nucleic acid coevolution, but that does not define life. You can put it in a protocell, that's not life. So the question of understanding origins of life itself is a struggle to understand the definition of life. Apart from that, when I was giving a lecture at University of Michigan, there was this associate editor of Chemical Reviews, Ruma Banerjee, who, after my lecture, we had a chat, and she said, Ram, you title your talk Origins of Life, but isn't it really origins of biomolecules? And I said, yes, we are only looking at origins of biomolecules, never at origins of life. But I told her that origins of life attracts more people, it's more sensational than origins of biomolecules. We have to this is the McDonald's approach. You sell toys, and then you sell food to the parents who come in. So they bring in their child for the toys, but then you make business by selling the food. So origins of life attracts a lot of people. Origins of biomolecules attracts only the biomolecular people. So it's a fascinating question. And as I think somebody, I couldn't find their name at that time because I didn't know who was talking. Kattadu kaivan alavu, kalladadu ulagalavu is always going to be true for us as it keeps expanding this literature. Anyway, thank you for the wonderful lecture. It was a simply superb walk into the memory lane to see, the triple helix. And I love the analogy that you had put in your paper of how he came up with the triple helix structure when he saw his wife braiding her hair. And it just reminded me of Triveni Sangamam. The Triveni, Veni is also in Sanskrit means hair, comes from that, there's a beautiful play in Sanskrit, Veni Samharam, about the death of Drona. The beautiful Triveni Sangamam came into my mind while I was reading your paper. Thank you so much for that.
That analogy was actually Ramachandran giving a lecture, and when I was a student, and that's what he said. I,
This is, can I make a comment?
yes.
Yeah, this is Birali Sashi. I attended, it's a wonderful lecture, and my background is medicine. And I was a student at the Indian Institute of Science from 1977 to 1980. I enrolled in your course for a week. And your favorite book was Mahler and Kodesh. And then I realized it may not be the cup of coffee, tea for me. So then I dropped out. But in any case, I really enjoyed your very informative and very engrossing lectures then and even now. And I have one comment about the origin of life from non-life to life. I just want to remind everyone of famous Miller's experiment, in which at high voltage or whatever energy, how biomolecules were produced from non-biomolecules, so to speak. You may comment further.
I will comment on this. I was about to say this in response to Dr. Krishnamurthy's long comment. It turns out that in 1953, two papers appeared. One appeared in Nature and the other one appeared in Science. The Nature paper was the Watson-Crick double helix of DNA. And the Science paper was Miller's Origins of Life paper. Only one of these was reported in the New York Times. And it is the New York Times headline, which says Origins of Life, which then associated Miller with the Origins of Life experiment always. The Watson-Crick double helix took a little while to sink into the scientific circles and then catch popular imagination. But the Miller experiment caught popular imagination immediately. I think it's just one of those things, but it was truly an origin of biomolecules experiment.
Yeah, that is exactly right, Balaram. I don't want to take too much of time here, but let us allow the younger generation to ask questions. The Miller experiment is one thing, and then there are deep sea thermal vents supposed to be another source of origin of biomolecules. And the third one is comet impacts. I'm working on comet impact. The problem is, as everyone elucidated, we know how the prebiotic evolution occurred, and we know how the biosystems evolved, but we do not know how the prebiotic molecules, whatever the macromolecules we can make, that primordial soup on Earth has transformed itself into a living system or systems, whether it is linear or non-linear, whatever it is. That is the biggest science gap we have in the knowledge base of humanity today.
Murthy, this is Arunan here. Can I just make a comment?
Yeah, sure.
Yeah, Balaram, excellent talk. But as someone mentioned about the experiment producing biomolecules from sparks, I must point out that neither explain biomolecules nor life. Both of them need chirality. And that is the real key now. So that Miller experiment was probably just a first attempt, but it does not really explain biomolecules or life. Just a comment.
Yeah, I might just, since people seem to be interested in this, you might look at work done by a person called Sutherland. It appeared either in Nature Chemistry or Nature Chemical Biology, I forget, not too long ago, 3-4 years ago. We're using very, very simple, hydrogen cyanide is a favorite starting material. Formaldehyde is another. These are all cometary molecules. And using these, you can make the DNA bases in laboratory experiments. You can put together sugars. And you almost, when you look at some of the figures in the paper, it looked like a page from biochemistry textbook. So it is fascinating. You throw in a few more things like hydrogen sulfide and copper, you get much more chemistry done. So prebiotic chemistry could have been very rich, well beyond Miller's experiment. But organization into cells is another problem.
For origins of chirality, if I'm not mistaken, it was just Stephen Meyer who did a lot of experiments, I think.
Origins of chirality, there are all kinds of theories, but one of the best is the amplification of a random event. Life is a random event.
Since Professor Balaram, we are talking about when did the origin of life versus origin of chemicals. There is a beautiful book which is called as What is Life? How Chemistry Becomes Biology by Prof. It's a wonderful book which actually gives, the gap that exists and how it is covered. So I would recommend this for those people who are really interested in this problem. Prof. What is life? How chemistry becomes biology. That's where the whole crux is.
I just want to mention Ram Narayan, who just asked a question before. He's at Scripps. He works in the area of origin of life. I think he's pretty well known on this topic.
I would say, actually, Ramurthy, I've read a lot of Dr. Krishnamurthy's work because Escher Moser was a co-author. I used to see whatever paper Escher Moser was then writing. And then I found that he changed his field from, I knew Escher Moser work when he was doing B12 synthesis. But then later the origin of ribose sugars, hexose nucleic acids and all became another area, which I thought was very interesting. Real fantastic stuff.
Anybody else has questions?
Yes, this is Murthy. I'm Bhagavatullah Murthy. I'm a professor of pediatrics at Baylor College. Hello, Professor Balaram. A very, very nice lecture. It was just fantastic. I don't have any specific questions on your talk. It was just very, very engaging. I just was telling my wife this morning that I attended your famous Balaram course in the 83, and then it was just, you would start at 9 in the morning and exactly finish at 10, and you would cover so much, and it was just amazing. I still remember, I can still, it's vibrating in my ear even now. So I just appreciate your teachings and your service to the institutes and for a lot of students for generations. Thank you.
Yeah, after 10 o'clock, the class finishes, you will go to the class.
Regarding the origin of life, I was wondering how did the first chiral molecule came into existence? So that is the fundamental question, right? So isn't it possible that in the beginning there was no chiral, there was no chiral molecule, everything was racemic, but one of the enantiomers reacted and the other didn't, and through evolution the enantiomer which reacted went along.
There are many detailed theories of the origin of chirality. One of them is amplification after an event where chiral molecule has reacted. There are others which deal with particles causing chiral selection. But there are any number of theories. Unfortunately, in the origin of life, it's easier to speculate than to find experimental act. And as you get older, it's natural to do more speculation and less experiment.
Yes, there is another theory that probably it didn't originate in Earth itself. Probably a comet, an asteroid, which had.
You see, that only puts the problem of origin of chiral chemistry elsewhere. It doesn't solve the problem. Hoyle's theory of panspermia is not doesn't solve this problem. That means it happened somewhere.
Yeah. So
the
if
question
this
would be,
is
how
the
did it
case,
happen
then that does not solve the problem. That is correct. But probably we should also look for chiral molecules in asteroids, meteors that have fallen down to Earth.
They have already looked, and NASA Goddard Research Center has documented that asteroid or meteoroid samples have enantiomeric excess. And we do not still know how this has happened, but as Balaram said, it puts still the problem. We got them here, but we don't know how individual molecules became self-governing, self-replicating systems.
Because since the asteroids, they are in space and you have gamma radiations, so I don't know whether gamma radiation can differentiate between two enantiomers, but if one enantiomer degrades at a faster rate, the rate constants of degradation of both the enantiomers is higher. Then one enantiomer will degrade faster than the other. So that might explain the enantiomeric excess that you are getting in asteroids.
My suggestion is, to come back to the subject matter of proteins and GNR and leave origins of life, because as Professor Balaram said, we are all speculating and really not really, taking up the spirit of the lecture that was given. So my humble request is to go back to the lecture that was given in terms of the substance and the inspiration that it has.
I have a question in that regard. My name is Rajesh Sundaresan. I'm a faculty member, current faculty member at IISB. I always wanted to know about Professor Ramachandran's research, and thanks for this fantastic lecture. I would like to know about Professor Ramachandran's approach to science and research. Was it one of a hobbyist, or was it somewhat more professional, Professor Balaram?
Yeah, Ramachandran was an extraordinary scientist in one way, that his focus was entirely on science. Nothing else. Nothing else bothered him.
Okay, so I think somebody has to turn off the mic. Or I have to turn off everybody's mic and then okay.
Oh, I think Ramachandran was an extraordinary scientist in that he was totally focused on science. He didn't make too much of small talk. He wasn't very much interested.
Who is this one now? Okay, I'm going to mute.
Can you please mute your mic?
Go to the lecture.
He's muted it. He's muted it now.
Okay.
He was an extraordinary scientist in that he was focused on science. He was not like he wasn't interested in very many other things. He wasn't interested in many things which people spend their time on. He liked to hear scientific discussions, and if you went to him with a discussion, he listened intently. He had another quality which I think is rare. I think if he was working on a problem, he saw the heart of the problem. Even if he didn't have a solution, he understood what the problem was. Many times we don't understand what the problem is, and we try to find a solution. So his ability to go to the heart of the matter was, I think, the key to his success. I also suspect that a few people do have this. I think he was able to visualize many things. He was able to think in 3 dimensions. We can now think in 3 dimensions with the help of a computer and graphics. But otherwise, just to sit and think in 3 dimensions is sometimes not quite so easy. There have been other people who could do that. And I think this was one of his advantages. And he was an expert in some areas of mathematical physics from his training in optics. Fourier transforms was one thing in which he was, I think, absolutely expert. He would sometimes pretend that he did not know things. I don't know whether he was pretending or he really forgot it.
Balaram, when I was there towards the end of my stay, he moved out of biophysics and went into a different department. And then mathematical physics or something. I forget what is the name of the department.
No, he went into what I would call local exile in the library.
Mm-hmm.
And called himself first, he called himself mathematical philosophy.
Mm.
First he said mathematical biology, then he said mathematical philosophy. And so in his sort of, when he entered these more difficult stages of his mental condition, he would suddenly focus on mathematics. He would work on problems, the great unsolved problems of mathematics. Those were aberrations, I think, which were brought on by his organic problems.
And then with respect to a couple of, because there are lots of students, we oversubscribed because our strength maximum is 300, but we had more than 300, so a lot of people are cut off after 300. And still there may be some students. So with respect to dealing with him as a student, was it easier or difficult, or he was encouraging, or what would you say? Or you might have seen these people struggling with him.
I've seen his students and I've seen his former students. All my senior colleagues were his former students.
Mm-hmm.
And all my senior colleagues were his former students till the very end, I would say. He was a professor. And I don't think it was difficult to work for him. He's always interested in your result. And if you got a good result, he knew what to do with it. He understood it. And he gave you good problems. I mean, what you want from a teacher is a person who will give you good problems and who would help. If you come with a solution, he would be happy. I think that his ability to recognize results was very, very good.
So what happens
when I was a young faculty member, he would recognize whatever result you got and he was interested in it. And the amazing thing was that if there was a committee visiting, he would present your result. It was obtained in the department. He would present your result. And when you listen to his presentation, you felt, look, he's probably presenting the result slightly better than you would have if you were asked to present it. So,
If I can,
I have some letters which I recently recovered from my old files when I shifted, and from him, handwritten ones, where you can't imagine a professor writing a letter to a lecturer like this, 2.5 pages by hand, and all of it is only about some conformational thing. Can you imagine anybody doing it today?
I have a small anecdote here. When I was doing my PhD with Professor Balaram, me and my lab mate decided to print T-shirts with the Ramachandran plot in them. And we just decided one day to go and present one T-shirt and a handkerchief to Professor Ramachandran. And after we did that, Professor Balaram came and asked, why did you do that? Are you going to go and see him? We said, yes. Then we had already written a letter and sent it to that guy in aerospace, I forget his name. And immediately we got a phone call from Professor Ramachandran, and he said, you can come at this specified time. So me and my lab mate went there with the T-shirt, and we were given a very nice coffee and some hot vadas. by Mrs. Ramachandran. And he was only speaking to us during that time. After he just saw it, he said, yeah, now the map looks correct, like this is it. And then he put it on the side, and he was only interested in what we were doing for our research, in our research problem. And after that, we used to get lots of requests for photocopies of papers from various journals, because he was a member of the Academy of Sciences and used to get the PNAS hard copy and used to go through every paper in it, I think. He was one of the rare guys. And then he used to get those back references from it and he used to send it to us. And initially we used to photocopy it at our cost and then on the lab cost. And eventually we decided that best way was to just stop it. But his interest and passion was really there to be seen. And I have not yet come across somebody who has a similar passion for everything that he reads. I mean, there are different aspects that we read. We like our own type of research, we like our fields, we like some historical things, and we like how people live their lives. But to have an interest in every scientific literature, I think you have to be beyond a genius. And that I think Professor Ramachandran was. I don't know if you remember it or not, but the t-shirt was very famous.
Did you sell any of those t-shirts afterwards, or just only one you made, or what?
No. We broke even. We broke even because thanks to India and UK, some symposium, we broke even. Otherwise, we were in a soup. We actually put in about 15,000 rupees, which was a lot of money in those days.
Oh.
We just broke even.
Okay. Okay, there are a few more questions.
Professor Balaram, very nice talk, very informative. Thanks. This is Prabhakaran here from Organic Chemistry. I was wondering, did Professor Ramachandran directly start with the van der Waals model, or were there other theories that he started with? How did he stumble, or how did he come up to that van der Waals clash model?
No, I mean, that's all that you can have with atoms. I mean, they were talking about contacts even then. You know that atoms are crudely approximated as hard spheres, and hard spheres can't come closer than a certain distance. The question is, are atoms really hard spheres, or can they be slightly soft? And what is the distance? The mistake that people had at that time was that originally probably the map, lots of structures would have been disallowed if you used the distances which were then prevailing in the literature. But Ramachandran's group, what they did was they looked at the few organic crystal structures which were available. And in organic crystals, looked at the distances to which atoms came. Those then became, from experiment, they became what were then called contact distances. So non-bonded contact distances were experimentally derived from crystal structures. But you must remember that the number of crystal structures, that is three-dimensional crystal structures, in the early 1960s, were very few. This was before direct methods came, and very few molecules, crystal structures were available. And then to get distances from those, you would have to take the coordinates which were there and calculate the distances again. You can imagine the kind of arithmetic task that would be. But they did it. They compiled it from there.
Thank you.
Professor Balaram, this is Sivaguru from Bowling Green State University. Excellent talk, very informative and educational. Can you comment on, the Ramachandran plot used for biomolecules? Can you comment on whether there are attempts or anything to do for synthetic macromolecules, because those also can feature hydrogen bonds, and then that might translate to dynamics or understanding of macromolecular structure itself. So can you comment on that? Thanks.
Yes, you see, for example, Ramachandran, immediately after the success with proteins, the Ramachandran group turned to polysaccharides and polynucleotides. So my senior colleagues, Professor Rao, V.S.R. Rao, used to work on polysaccharides. So if you take a disaccharide, it's connected by the glycosidic bond, and there are two torsion anchors. Similarly, if you take nucleic acid, once in the biopolymers, interestingly, the rigid units are connected by two rotatable bonds. and the phosphodiester. And so you can actually do such work. But in the case of the nucleic acids, there are very much more constraints because of hydrogen bonding. So that problem is a somewhat different problem. Whereas in the case of oligosaccharides, polysaccharides, it's an ongoing problem. There's a fair amount of...
how about synthetic macromolecules, like urethanes, for example? So that feature...
in principle, yes. The problem is this, that if you have between each monomeric unit 2 rotatable bonds, you can use the Ramachandran map, the Ramachandran type map. That's what I tried to illustrate for bilirubin, for instance. But if you have more than 2 torsion angles, then of course the problem becomes a little bit more complicated. You won't get such an easy representation. But you
is it because that you have multiple planes? Is that because that you have...
Yeah, you have multiple planes. You have multiple torsion angles. Let's say you have 3 torsion angles. Then it becomes a 3-dimensional.
Yeah, instead of 2-dimensional, you have to have the third axis then.
You take 2-dimensional sections of it. In principle, you can do a 4-dimensional map too by taking sections. But it's very it's not elegant, and it's not...
No, but I was wondering whether it can give insights into, like macromolecule structure itself, because that determines the property of the macromolecule.
Absolutely, because for example, in the field of polypeptides, where people have gone on to make polybeta peptides, polygamma peptides and all, and not polyalpha peptides, which are proteins, you have this extra torsion angle problem. But it doesn't get sorted out as easily as... But what happens is, if you have CC bonds coming, there's a simplifying factor that, every organic chemist knows that there are 3 staggered conformations for about the CC bond. So you limit that, and then you can go back to a 2-dimensional. You could do that. You could have multiple 2-dimensional.
Okay, great. Thank you.
I just want to add there, Professor Balaram showed this macrocycle example where it was applied. That was a pertinent one, Sivaguru, where he showed that example from Nature Chemistry, where he showed how they actually used the Ramachandran plot there, and it came out very nicely. So that might be another example.
Okay. Okay, thank you.
Professor Balaram, can I ask another question? It's more general in terms of, GNR's legacy, and also what happened since then, and how we are heading in India in terms of scientific culture and scientific progress, right? If I go back to 1980s, and when I started my PhD, right? When we look at the publications in JACS or any of the top journals, it is completely not a very objective analysis. But I remember that we used to have from India many more number of publications compared to, say, in China. But now I see that every journal, every top journal will have top level publication from China, and we have stagnated in the same, maybe it's harsh, my wrong assessment, but India stagnated and stayed almost in the same place as in the 80s in terms of number of publications in top journals. Do you think that is a correct assessment, or we are...
No, India has not stagnated. India is going along steadily. China, which has exploded. There's a difference. For that, you have to study China rather than worry about India. You have to ask the question, if today the United States is worried, I read a report that the United States now, they're trying to ask for $200 billion or something to be put into the scientific enterprise to make sure that you stay far ahead of China. That's a different matter. I don't think one should be comparing India and China in that way. One needs to understand China, and China is very difficult to understand.
Great. I mean, there is definitely more investment, right? They become richer and then more investment. But it's not just the money, but they also build these institutions, right? Where scientific, instead of being a communist society, they invited outsiders to come and start institutions. India, there is always this resistance to inviting outsiders and cross-fertilize, these ideas, right? India, if you're in state of Andhra Pradesh, only Andhra Pradesh people can become professors in those institutions, right? Is that played into our less kind of progress in addition to not having money?
I don't know. I think this is a general question. It's one that addresses to governmental policy and so on. One of the things that, at least in a Ramachandran lecture, I would say is Ramachandran and government were pretty far apart. He was a student of Raman, and I think he had inherited many qualities from him, probably also, in addition to his own.
No, is exceptional, right? It definitely, as somebody said, he's a genius, right? So and he actually drove a lot of us into that culture, right? And we benefited it directly or indirectly, right? But only one person can take you so far, right? So everybody else following him should have contributed to this progress. But I would, as I said, it's a harsh assessment saying that we stagnated. We did not, but we did not, progress to the highest potential we have with so limited, resources. G.N. Ramachandran or many people I can quote at that time, right? They've made such huge, impact on world science, right? And that legacy we failed to carry on.
So that actually I would argue with. In fact, if you look at the United States, for instance, fields grow, and after some time, it's not possible to find out in fields which are truly fundamental advances in a field, because science grows at an exponential pace. Since many of the people listening here are organic chemists, I would ask, is there the kind of excitement there is in organic chemistry that there was when Woodward and Corey were at their prime? The answer is no. Similarly, if you go back and look in other fields, America also has many scientists who we still celebrate. It becomes harder and harder to find that kind of impact when science has grown enormously. It's also a group activity now. Yeah. It's an activity which involves huge amounts of investment, organization, and so on. The individual scientist is rare now. And it's becoming rarer and rarer for an individual scientist to make an impact. In Pasteur's time, Pasteur could be the founding father of microbiology and the founding father of stereochemistry at the same time. In the 20th century, nobody could be the founding father of two completely diverse disciplines. In the 21st, it's going to be even more difficult.
Yeah, it's the time for these group activities, or you have to be a Sabyasachi or ambidextrous, right? You have to have multiple skills to function in this. You have to be a data scientist and also computational biologist, also molecular biologist, right? So to make an impact, you have to have multiple expertise. You have your own, or you bring those people together to solve a bigger problem. You can single-handedly, you cannot solve these problems, right? So in terms of organizing ourselves into these groups, working without these jealousies and competitions, coming and to work for a larger mission is that kind of organization. You said that is the organization skills. Did we really develop those skills to come together and take on bigger challenges and work together? That, I see it here, constantly people working together on many big things. We come from.
We are very close to 10 o'clock over there.
Can I make one comment? Can I make one comment?
Sure.
Birali Sashi. I think there is a fundamental difference between 50 years ago and now. 50 years ago or even before, there are many outstanding problems staring at you. And so whoever could pick on those problems and solve, that's one thing. Currently, all the major outstanding problems, not to mention physics and other fields in biology, like genetic code, like DNA structure, like immunoglobulin structure and gene structure, they were all solved. Now it's only trying to understand more details, and moreover, the scientist has to define the problem. Previously, the problem was already defined. It's a matter of solving. I think that's one important difference between why we can't have stars now as opposed to in previous days. For example, in Newton's time, whatever he discovered, it was the whether it's a calculus or a gravitation or whatever. So if you trace from that time to this time, now most of the problems are solved. But of course, now it is the responsibility or ingenuity of the scientist to define the problem, isolate the problem, then solve it. So it's an additional difficulty is there.
Balaram, we should be finishing very soon. I want to ask whether there are any students with questions. I guess they don't have any questions, or there are no students.
There should be students.
So we are all students. We are all still students of for science. I have one question. If nobody else has, this is going back to the collagen problem that Professor Balaram pointed out. I would like to get more understanding into how Professor Ramachandran was affected by that he had to leave or decided to leave the field of or not touch collagen anymore. But because in your article about him, you mentioned the first time you met him, he asked you whether you knew about collagen, and you said you didn't, and he was kind of disappointed with that.
He was interested still in collagen even when he came to Bangalore. One of my colleagues, Professor Manju Bansal, actually worked with him for her PhD on collagen. But by this time, collagen, it was only detailed.
Okay.
The broad features of the structure and everything had been accepted, and there wasn't that much of interest at that time in the 70s on collagen. Collagen interest revived later again when people began to find mutations in collagen genes, relationships to disease, and so on. And now you began to worry about what were the effect of mutations on the collagen structure, the melting temperatures of the collagen helix, and so on. So the collagen field rebounded. But by that time, Ramachandran career was more or less.
Okay, thank you.
So,
he had a relatively short career, even though he started early.
Balaram, can you probably say something what, in your perspective, life of Ramachandran, could inspire and do's and don'ts to the aspiring or present time budding scientists, not the old folks like us. We are already at the later part of our career, but those who are budding scientists, what is your advice to them based on Ramachandran's life?
I don't think one can give students and young scientists any advice. Actually, it's wise never to give advice because advice is generally disregarded. But sometimes reading about scientists and their work can be inspirational. See, if you look at biochemists, biologists, you'll find any number of them have been inspired when they were young, the famous ones, by a book which appeared at that time by Paul de Greve called The Microbe Hunters. Because at that time it described Pasteur, Koch, and everything, and people were inspired and they went into biochemistry, microbiology, and things. Whether it's Kornberg or many others, many people have written this. Many physicists who went into biology read Schrodinger's book, What is Life? And so there are always these sorts of general reading that one does. Today you can read a lot because of the internet. And some of the stories are truly inspirational, even for old people. You sometimes think maybe you should have read the story 20 years ago, 30 years ago. And you also sometimes think, okay, maybe it's not too late. Okay. So I would guess that people should read about how things have actually happened.
I know, I'm not sure. There is one question I see, but that may be a student or may not be a student. I will read the question, and maybe with that we can probably wrap up, unless there is you want to continue to discuss about how to change things. Okay, so here there is a question from Kavya Shri. Thank you for a brilliant and informative talk. Sir, if there is one thing that need to change in India which would encourage and promote science and scientists more, what would it be? Only one. So she's asking.
I think I am the wrong person to ask this question too, because at the present stage of my career, maybe I'm not thinking straight about how to improve science.
Okay.
And what is it that should be done? See, people like me who've been around for a long time in India and who have seen many things, we've become used to actually doing whatever we can, not necessarily what should be done or what one really wants to do. I'll tell you one story about to conclude all the stories that I've been telling you. This is about Ramachandran. In our department, one of the things is one of my colleagues, Professor Vijayan, who came from Dorothy Hodgkin's lab, he had the ambition of setting up a laboratory for macromolecular crystallography. But there was no equipment there to do any macromolecular crystallography. And more importantly, there were no crystals of any proteins to do anything with. There was one old X-ray generator and an old Weisenberg camera which was available. So the first thing that we wanted to do in the institute was at least solve some structures. So one would get, the first thing was maybe could one get an X-ray diffractometer? But the institute at that time did not have an X-ray diffractometer. So they began to try and get money for an X-ray diffractometer. So after all this was done, you couldn't get a diffractometer because there were a couple of instruments like this for which you couldn't get because you needed import licenses. And you needed other clearances which would precede getting the import license. So finally, one day, Ramachandran went to Delhi carrying all the documents with him. Now, he, of course, I would say was clueless about what should be done in Delhi. But because of some connection, he managed to reach the office of an important bureaucrat. And he gave him all these documents and then broke down and saying that can't do anything without these things. I think the gentleman on the other side of the table was so alarmed that he decided to use whatever influence he had to get Ramachandran those clearances almost immediately and gave it to him so that he could get him out of his room. And I still remember Ramachandran coming back very triumphantly with these papers. See, I got them in one day. Now, that was the kind of situation which was there in the 1970s when he was going out. And many of us were just starting. So today, many things have happened. So if you want to ask the question, what is a single thing should be done in India? I don't think there's any single thing that will do anything. Many things have to be done, little things. And many different people have to do them.
Sir, this is Jaimurugan. Can I ask a question?
Okay, this be related to GNR and then.
Yes, GNR.
Yeah, okay.
Actually, yeah, I would like to learn your opinion on some of the controversial points. For example, Watson Creek structure, Nobel Prize work might have been given to Professor Dorothy Atkin because she was originally solved the structure, something like that. And similarly, now Professor Katha also wrote letter to Professor G. N. Ramachandran that the paper is getting delayed purposefully. So that kind of controversy, I just want to know from you, what is the actual scenario happen?
No, I know only what is there in the letter, because I don't know what actually happened. But Kartha's letter clearly says this was talked about a lot, and it's not unusual. Look, it was easy to publish papers in Nature if you were in Cambridge. And you read Watson's book. You see, the old editor of Nature, Richard Gregory, used to go to a club in London with all the manuscripts stuffed in the pockets of his greatcoat. The story has it that he would pass it around to the club members who would be drinking there and get an opinion on it and then publish it in the next issue. Those things were then, not now. So,
okay, thank you, sir.
I think as Professor Balaram was saying, some of these things, if you read some biography of some of these great scientists, you might be able to get some insight into the difficulties people go through. Even Einstein, for example, he had to struggle to get a job. He was tutoring people, and he was putting advertisement in the papers. And then finally he got a job at the university, and then his course had 3 people. And then eventually only one guy showed up. And then he himself got tired of teaching one person and then he quit. So he had, everybody goes through lots of problems, to be successful. You cannot expect everything is going to go smooth.
But I was wondering, sir, so that time Linus Pauling, Crick was in competitors. And the paper goes to them and then they delay it. Why not you might have just published in Indian journal, some lowest journal, impact journal, because the work will not get low regardless of where it get published, to at least to climb the first work.
Yes, I think Ramachandran did publish some of his work. For example, he did some original work on anomalous dispersion around the same time as Barefoot, and that appeared in Current Science. One of his collagen papers also appeared in Current Science. I had put the reference here. So that was done. But, publishing, recognition, credit, these are all something different from actual work itself. We cannot actually rationalize why something happened and something does not happen. In retrospect, we can give Ramachandran the credit. If other people did not give it to him at that time, it's one of those things that happens in life.
Yeah, can I put a quick plug on Indian Institute of Science Alumni Association? We have in America, Indian Institute of Science Alumni Association of North America, and we are working with IISc. If you want to work with us, it is a free membership. Join us. A lot of us here are cherishing the memories of IISc, and we would like to give back. Join us, and I put that in the text, in the chat. That's my plug. And I think, as Balaram said, I think it is important to not put the person in the front rather than science. Let us learn that it is not about our ego. It is about advancement of science. And humans, by definition, irrespective of where we are, we have limitations and we have mistakes made. But we cannot just hang on to the mistakes and say that everything is unfair around me. Keep marching, keep doing good work. You will make it. Sorry, Balaram, for that monologue.
Well said.
Well, you plug the needed info about your alumni association. So hopefully you'll get some more people. Balaram, I just want to mention to you, I see Professor Venkateshan's wife. She is actually listening to your talk.
Oh boy.
Yeah.
Oh, is mommy there?
Yeah, I think Raji.
Oh, Raji, wonderful.
Yeah, oh, that's logged on her name. Yeah. She
Yes, we are both here.
Okay.
Let me just get her on video. Yes, we are both
okay.
here.
Raji, can you put your contact in the text, in the chat?
Yes, I will certainly do that. We really enjoyed the talk, Professor Balaram, and thank you so much.
And Murthy also is there. Murthy is Professor KV student.
Germany Murthy.
Germany Murthy மேல உட்கார்ந்திருக்காங்க.
I will put my contact.
Okay. Yeah. All right. So we will close. So thank you.
சொல்ல திருப்பி என்ன ரொம்ப நன்னா நடத்திட்டேங்கிற
அவர் தானே நடத்திட்டு இருந்தாரு. என்னோட வேலை ஒண்ணும் இல்லை. Okay, so thank you, Balaram. Nice of you to have agreed to give a talk in this forum, and we had the maximum crowd. I didn't realize it. Our number is only 300, so after 300 we had problems. So excellent talk and a lot of good advice to people. Although you don't want to give advice, we will take a lot of advice from your talk. All right. Okay. Thank you. Thank you, everybody.
Have a good day. Bye.
Thank you.
Right. Thank you. Bye, Balaram.
Bye.
Thank you.
Bye. I'll be in touch. Bye.
VR, can you save the chat?
Say the what?
Chat, the chat files. Probably
how do you save that? I don't know. Many things I don't know here.
There is an option to save it. If you go to the chat.
Charts are automatically saved in the cloud along with the video.
Yeah, the cloud. I think it actually prints out a script, right?
Yeah. So that has
towards the corner there are three dots where you can do save chat.
Yeah.
Okay. All right. I think first I will stop the recording.